Hello students, the topic is biotechnology and its application, and in today's class let us learn how pest-resistant plants are produced by using a novel method that is called RNA interference. RNA interference was a new mechanism which was used by biotechnologists to protect Melodegni ecognitia infection in tobacco plants. Melodegni ecognitia is a nematode. Nematodes are roundworms. Just like how roundworms cause infection in human intestine, there are nematodes which infect certain plants. In this case, Melodegni ecognitia is responsible for causing infection of the roots in tobacco plants. So when tobacco plants are infected by Melodegni ecognitia, the infection of the nematode causes formation of tumors. These tumors result in reduced productivity in the tobacco plants. So scientists thought of using a novel mechanism to prevent this infection by nematode, and this mechanism was called as RNA interference. So first let us understand what is RNA interference. The RNA interference is a mechanism which is seen naturally in many eukaryotic organisms including plants. This is a mechanism by which the plant defends itself from certain infectious agents like RNA viruses. The principle of RNA interference is based on silencing of the mRNA. When we say silencing of the mRNA, means it is a process by which the mRNA becomes non-functional. And non-functional mRNA will be unable to produce the specific proteins. And in absence of production of specific proteins, that means when mRNA is not translated into proteins, the infectious agent is unable to cause infection in the host. Now this happens because in natural conditions, a double-stranded RNA is introduced inside the eukaryotic organism like the plant. So as I told you, this may happen when RNA viruses infect the plants, or it could happen because of mobile elements called as the transposon. As soon as this double-stranded RNA infects the plant's defense mechanism is activated, and this defense mechanism is called as RNA interference or RNAi. Now, this RNAi activation results in production of certain proteins, and because of which the RNA becomes non-functional. That is, the mRNA of the virus or that agent which has infected the plant becomes non-functional. And when the mRNA is non-functional, the infection is stopped at that stage itself. So now this is what happens in eukaryotic plants or organisms as a natural defense mechanism. Now the scientists tried to use this natural defense mechanism, which is called as the RNA interference, to protect the tobacco plants from the infection of Melodegni ecognitia. Now let us see how exactly they did this. The scientists introduced the double-stranded RNA specific to the nematode. That means this double-stranded RNA which was introduced inside the plant was a nematode-specific RNA. Now this RNA was introduced artificially inside the plants, that is the tobacco plants, using agrobacterium vectors. So that means a double-stranded RNA of the nematode Melodegni ecognitia was introduced in the tobacco plants using agrobacterium vectors. Now you know students that agrobacterium is a bacteria which is responsible for causing tumors in plants. But in agrobacterium vector, the tumor causing genes are now removed and the specific genes are introduced. In this case, the specific genes were the double-stranded RNA of the nematode Melodagny ecognitia. So as soon as this double-stranded DNA entered inside the plant system, the mechanism of RNAi was initiated. The textbook does not mention what exactly happens in RNAi, but in the following video you will learn about it, that RNAi actually involves formation of specific proteins called as dicers and another type of protein integrated complex which is called as RISC. This entire mechanism of formation of RISC and dicer is called as RNAi or RNA interference. So in RNAi what happens is that ultimately the mRNA which is going to produce a specific protein of the nematode which is responsible for causing infection in the plant and responsible for causing the tumorous growth in plant is not allowed to get translated because the mRNA gets bound by these specific proteins. Now these specific proteins produce a single-stranded RNA. This single-stranded RNA binds to the mRNA, thus forming a double-stranded RNA. And this double-stranded RNA is said to be silenced as it is unable to cause formation of proteins, thus preventing the infection at that stage itself. So you can see in this slide, students, that the green, what is shown here, rectangular figure here, represents a plant cell, and the orange figure represents the nematode. So you can see that a double-stranded RNA is introduced inside the plant cell. Now, as I told you, when RNA interference mechanism is initiated, what happens is that certain proteins are produced. These proteins are in the form of dicers. Now, these will cleave this double-stranded RNA, this foreign double-stranded RNA which is introduced inside the cell, either naturally or using the agrobacterium vectors, is broken into small RNAs which then join with some other proteins to form a factor called as RISC complex. This RISC complex then cleaves these small RNAs into single-stranded RNAs which are shown in red here, and these are going to then bind to the specific target mRNA. So what is going to happen here is that when the target RNA is bound, this target RNA becomes non-functional and it is unable to produce the proteins. So you can see here this double-stranded RNA is introduced inside the cell. This initiates the RNAi or RNA interference mechanism, which involves formation of this dicer and this RISC complex. This is not mentioned in your textbook, students, but this entire phase is called as RNA interference. So the RNA interference defense mechanism is initiated, because of which the mRNA, the specific mRNA, gets bound to this RISC complex which contains the single-stranded RNA. As a double-stranded RNA is formed here and double-stranded mRNA is not functional, this further gets broken down, and as a result, because mRNA is no longer functional now, it deteriorates or it gets degenerated, it is unable to produce the proteins, and this is called as RNA silencing or mRNA silencing. So you can very well see this in this video.
RNA interference is a mechanism of silencing gene expression after the gene has been transcribed. This interference is due to the presence of antisense RNA, which is complementary to a specific cellular mRNA. The complementarity between the antisense RNA and the cellular mRNA causes the two molecules to bind to each other. The antisense RNA can come from three sources. First, it can be introduced into cells by researchers. Second, certain viruses may produce antisense RNA as part of their life cycle. And third, the integration of genes into chromosomes next to endogenous gene promoters can sometimes cause the synthesis of the antisense strand of RNA. The current model of RNA interference involves two steps: creation of an RNA-induced silencing complex, RISC, and then the degradation of cellular mRNA by the RISC. Creation of the RISC occurs when double-stranded RNA is found in the cell. This double-stranded RNA is recognized by an endonuclease known as dicer that cuts it into pieces that are 21 to 23 nucleotides long. An antisense RNA fragment can then associate with proteins to form the RISC. The silencing complex can then associate with cellular messenger RNA that has a region of sequence homology with the RNA fragment in the RISC. The messenger RNA is then degraded. The presence of this rapid degradation mechanism can reduce and even effectively eliminate translation of the messenger RNA for the gene in question, thus interfering with gene expression.
So students, I hope you have understood the mechanism of RNA interference, which was very effectively used to protect tobacco plants from infection by a nematode called Melodagny ecognacia. Thus, we have studied about RNA interference as a mechanism to produce pest-resistant plants. Thank you, students.